My name is Sakshi Kankar, and today I am going to explain about the topic signal detection. Signal detection is in a pharmacovigilance is the process of identifying potential safety concerns related to the drugs or medical products based on data from various sources. These signals may indicate a new adverse drug reaction or a change in frequency or a severity of known adverse drug reaction. Then we will see what is the definition of signal. uh, a signal is defined as the information suggesting to a possible causal relationship between drug and adverse drug event, which is either new or previously known, but requires more further evaluation. Then there are few sources of data for signal detection. First is spontaneous reporting system. uh, the adverse drug event, adverse drug reactions report from healthcare professionals, patients, or pharmaceutical companies is collected for the data of signal detection. Then clinical trial data is collected. Safety data collected during the pre-market and post-market clinical trials. Then there are electronic health records which are studied for the data collection of signal detection. The real world data from patient's medical record is collected from electronic health records. Then there are literature and case studies which are studied for signal data for signal detection. Reports from scientific journals and case studies. Then there is social media reports where a who are reported outcomes and discussions on social platforms are collected. Then there are few methods for signal detection. First method is qualitative method. Qualitative method includes the expert review of ICSRs and literature. And then there is quantitative method. Quantitative method includes this proportionality analysis. It measures the observed versus unexpected frequency of ADR. uh, ADR, reporting odds ratio, or proportional reporting ratio. Then further is Bayesian data mining, which uses the Bayesian inference to identify the potential signals. That is then time to signal analysis is studied, which examines the time of onset of adverse drug reaction. Then advanced tools are used for like machine learning, which has pattern to recognition and prediction. Then there are few processes of signal detection. First is data collection. Data collection is aggregating the safety data from various sources. Then there is data cleaning, which addresses the incomplete or duplicate reports from collected data. Then there is data identification. Identification applies to the statistical tools or a manual review or to identify unusual patterns or trends. Then there is validation. Then validation accesses the mm, false information of the identified signal using existing evidence, literature, and expert judgment. Then fifth is signal prioritization. Prioritize the signal based on severity, frequency, and potential impact on health, public health. Then signal assessment is investigation of causality using established frameworks like WHO, UMC causality assessment, or Bradford Hill criteria. Then there are regulatory actions taken. If the signal is confirmed, regulatory agencies may update the product labeling, issue safety warnings, or recommend the further studies for that product. Then there are further challenges of signal detection. First is under-reporting of ADRs, then poor quality or incomplete reports. Then there are confounding factors like comorbidities or polypharmacy. Then there are high false positive and false negative rates of ADRs. Then there are few regulatory bodies and guidelines included in signal detection. First is World Health Organization, WHO, for international drug monitoring, WHO program. Then there is US FDA, which guides the sentinel intensive or active signal detection. Then EMA, which has ultra vigilance systems for signal detection. Then Indian Pharmacopoeia Commission, which includes the pharmacovigilance program of India. Then the signal detection plays a crucial role in ensuring the patient safety and improving the risk benefit profile for medicines. Thank you.